Hey guys, my name is Max and welcome to Simple Biology. Today we're going to be introducing the cells unit. Now the cell is an incredibly fundamental unit of biology. It's the smallest piece of life that can still be considered alive. In fact, every single organism we've come across to date, from humans to plants to fungi to even to bacteria, every single one of them has cells, or maybe sometimes just one cell. But that's why it's going to be so important to know how cells work and what they look like. In this video, we're just going to go over kind of a brief overview of different parts of the cell and how they work before we get into all the detail throughout the unit. So that's what we're going to be doing right now. First, we're going to start with animal cells. Now, oftentimes we're most interested in animal cells because, as you well know, we are animals. Some of the most prominent features that we're going to be looking at are in this diagram right here. The first one is the nucleus. Now, oftentimes, the nucleus is the only thing in the cell that you can see. It's the largest and most prominent feature we have, and it's also incredibly important. So that's why we're going to start with it. There's a couple different parts of the nucleus. The first we're going to look at is the nuclear envelope, or nuclear membrane. It's actually made of two separate layers of membrane, two different membranes, and so that's pretty important, and it has a role in its function. And within that envelope or membrane, you have just the rest of the nucleus. That membrane, that double membrane, that envelope, allows the contents of the nucleus to be separated off and kept together in order to play its role more effectively. Within the nucleus, you also have a nucleolus, or multiple nucleoli. This is just a region within the nucleus, and it's where ribosomes are made. We'll talk about ribosomes in just a minute. In addition to the nucleoli, or nucleolus, and the nuclear envelope, you also have a massive chromatin. Now, chromatin is what we call DNA when it's just kind of loosely uncoiled, and it can be used for its function when it's in this form. So you just have chromatin lying around. There are also these little pores in the nuclear envelope throughout the nuclear envelope, and those allow different materials to be exchanged between the nucleus and the rest of the cell. The next thing that we're going to look at is what's known as the endomembrane system. And this is basically just all the other membranes that are inside the cell, and so that's what we're going to be looking at next. The first thing is the smooth endoplasmic reticulum. Now, the endoplasmic reticulum is just kind of a maze-like web of membrane space, and it's actually continuous with the inside of the nuclear envelope. The smooth ER, or the smooth endoplasmic reticulum, or ER, is where some different things are manufactured, are produced in the cell. Often these will then be exported outside of the cell. The next part, there's also another part of the endoplasmic reticulum known as the rough ER. And the rough ER is different because it has ribosomes embedded on it. Now, I mentioned ribosomes earlier. Ribosomes essentially manufacture proteins. That's their function. So the rough ER plays a similar function to the smooth ER, but the things that it manufactures, that it creates, are proteins rather than just anything. Next, we have the Golgi apparatus, or Golgi body. And the Golgi body is actually, it looks like a big stack of pancakes. It's just these flattened membrane sacs, just kind of stacked on top of each other, and it's actually the cell's packing and shipping center. So after those materials are made in the ER, they'll be exported over to the Golgi apparatus before being transferred out of the cell or to different places within the cell. Next, we have what are known as vesicles. Now, vesicles are just little bubbles of membrane, and they're created by the Golgi apparatus in order to hold those different materials that were made in the ER. Another member of the endomembrane system is a vacuole. Now, a vacuole mainly just holds water in different solutes, and it looks like this little tan color up here. An animal cell will have many different vacuoles just kind of around the cell. There's only one shown right here just for simplicity, and there's a different type of vacuole that we'll be getting into next with plant cells. The last couple members of the endomembrane system are lysosomes, which look like this little purple dot, and peroxisomes, which are this little like bluish dot. Now, lysosomes and peroxisomes essentially digest different materials that are in the cell. They have enzymes within them that will just break those different materials down. And so that's what they do. Next, we're going to be looking at the cytoskeleton. Now, the cytoskeleton is all centered around these two little pieces of material, and this organelle is known as a centrosome. The two individual pieces are known as centrioles, and they're just oriented at right angles to each other, so two centrioles make up a centrosome. Radiating from the centrosome are these microtubules, and they're these little yellow lines, and microtubules are also found throughout the cell, but they're organized by the centrosome. So in addition to those microtubules, we also have intermediate filaments, which are these like blue kind of sweeping lines, and microfilaments, which may be a little bit hard to see, but they're little brown lines that are right there. And so all of these pieces of the cytoskeleton are found throughout the cell, and they hold the cell together, hold it in the right shape. Next, we have the cell's flagellum. And if it has more than one, then they're known as flagella. Now, the flagella are important for movement, for helping the cell move from one area to another. Not all animal cells will have flagella, but this is what they would look like if they did. Next, we have the mitochondria, which are where cellular respiration happens. That's known as the powerhouse of the cell. It takes glucose and other materials and uses them to give the cell energy, and so that's what these mitochondria look like. Next, we have the cell membrane, which just kind of surrounds the cell, holds it all in one place, and these little finger-like projections from the cell are known as microvilli. And what these do is they increase the surface area of the cell in order to maybe pick up nutrients from outside of the cell. Next, we're going to move on to plant cells. Now, plant cells have almost all of these organelles in common, but we're just going to go into the ones that are new, the ones that animals don't have. So, obviously, one of the most significant is the chloroplast. Hopefully, you've heard of a chloroplast before, but a chloroplast is where photosynthesis happens. It's it's green. It's what makes leaves and other plant cells green. So chloroplasts are incredibly important for plants. We also have the cell wall, which animals don't have. Certain other organisms in life have cell walls as well. Most significantly for us are the plant cells that have these cell walls. The cell wall essentially provides further structure for the plant cells in order to hold them in the right shape. However, because the cell wall is there, the cell has to have these little pores known as plasmodesmata that go across the two walls of adjacent cells in order to allow them to exchange nutrients. Lastly, you have the central vacuole. I mentioned vacuoles with the animal cells, but the plant cell will only have one vacuole, but it's huge, it's massive compared to those animal vacuoles, and it plays a role in the growth for plant cells, as well as storing water and different solutes for the plant cells as well. So now that we've talked about plant and animal cells and how they're different, it's time to talk about one aspect of cells that plays a role in both animal and plant cells, as well as all cells in general. And that aspect is the cell's size. Now, the important thing to know is that for a cell to get nutrients, the cell's nutrition, it depends on membrane area. So the cell can only take in as many nutrients, or as much nutrient as it has surface area of its membrane. Now, you also have to realize that its needs, how much nutrients it needs, is determined by the volume of the cell. As the volume of the cell increases, the cell's surface area doesn't increase at the same rate. That's the big thing to know. Obviously, volume is a cubed dimension. Area is a squared dimension. That plays a role. There's a math behind it. We don't really care, but know that a cell needs to be small enough that it can get the right nutrients for its volume. Because of this, the proper surface area to volume ratio is necessary. It's not so much how the size of the cell, it's not so much whether it has the area or whether it has the volume, it's the ratio between the two of them that is important. Now, because it's only that ratio, a cell has different ways that it can actually increase its surface area without increasing its volume very much. If you remember from the animal cell diagram, animal cells can sometimes have microvilli. Now, microvilli will increase the surface area fairly significantly without increasing the volume much at all. Another way for a cell to do this is to have a long, stretched out shape, and this will allow the cell to increase its surface area without increasing its volume much at all. And so that's really it for now. Check out all the rest of our videos in this unit, but the intro is as simple as that.